Hello everyone. Today we're going to be finishing up our second lecture on chemistry when it comes to human physiology. Today we'll be talking about organic compounds, which are compounds with a carbon backbone. And so many of the molecules found in our cells are organic molecules and have that carbon backbone. And attached to those carbon backbones are many different functional groups. And these functional groups will be used to make different molecules within our bodies. Some will be cleaved off for energy and to make other types of molecules. But here you see them listed in these tables here. Hydroxyl groups and O and H, carbonyls. So these are all structures that you should memorize. So when they come up in future chapters and in future lectures, you know what we're talking about, because many of them are very important for certain functions that occur within our cells, such as phosphate, which are broken off from a molecule called adenosine triphosphate for energy, aminos, which are attached to amino acids, the building blocks for proteins. So it's very good just to study these functional groups. This way you know what we are discussing when we talk about cellular reactions later on. First, we're going to be discussing carbohydrates. And I know in health and fitness, carbohydrates sometimes get a bad rap. I will say that they do have their place depending on what someone's goals are. However, we are just going to be talking about it in the purely functional sense, what they are, how our body uses them. And so carbohydrates are an important fuel source. A lot of cells specifically just use carbohydrates for their fuel source, including the nervous system. So they come in all different sizes and shapes. And so, a monosaccharide is going to be a single carbohydrate monomer. A disaccharide is going to be two of those, a dimer, and then polysaccharides is anything more than two of those monomers put together. Here we see examples of monomers or monosaccharides for carbohydrates. Up top we see those pentoses, those 5 carbons, 5-carbon backbone to form that pentagon there. So deoxyribose and ribose are their names. If you don't know those, that's okay, but just letting you know that deoxyribose is the sugar that is attached to DNA, and ribose is the sugar attached to RNA. And we'll be discussing in future chapters the role of DNA and RNA within our cells. Down below, you see probably more familiar monomers when it comes to carbohydrates, that being glucose, fructose, and galactose. And so these are going to be building blocks for different dimers and polymers for carbohydrates. So here we see how we can combine monosaccharides to make dimers and polymers, or disaccharides and polysaccharides. So up top, we see the combination of glucose and fructose, the binding of these two monomers together to get sucrose. And this is a very important time to stop and talk about, sort of the chemical reaction taking place here. And you can notice it's going both in the forward direction, adding these two monosaccharides together, and then in the reverse direction, the breaking apart of a disaccharide here. So the anabolisms, the anabolic reaction, the building up of these two monomers together, that's going to be done through something called dehydration synthesis. And so why is it called that? We're going to take a look at the rings here and notice the hydroxyl group, the O and the H on both sides. Well, enzymes a lot of times are going to bring these molecules in close proximity and catalyze these reactions to, get rid of 2 hydrogens and an oxygen. So we're dehydrating, we're getting rid of the water. And then a bond is going to be formed between the 2 monosaccharides right here with this oxygen. And so now, because we're getting rid of the water, the 2 hydrogens and the oxygen, we get the synthesis of a dimer through that dehydration, right? But it also works in the reverse direction as well. Water is used a lot of times to cleave apart dimers and polysaccharides. So here you see in the reverse direction that water can be used to cleave apart this bond, and then we're going to be left with the glucose and fructose. And so it's not just carbohydrates that go through these processes. Many different enzymes are used to bring or bind together proteins and DNA and all these different organic molecules to either, again, build them up through dehydration synthesis or break them down through hydrolysis. Down below, you see the combination just doesn't need to be glucose and fructose. Galactose and glucose can be bound together to make lactose. And so this, as you know, is found in many dairy products. Could also be cut apart as well through an enzyme called lactase. And, you may have heard of lactose intolerance, and that has to do with the ability to break apart this galactose and glucose from each other, which is why sometimes those with lactose intolerance, have issues once consuming dairy products. And then down below, you see the combination of glucose, two glucoses to make maltose. So we can add multiple monomers, multiple monosaccharides together to get polysaccharides. And here you are seeing, not just two put together, but we're adding on chains of glucoses in order to store them in our bodies. And this molecule, or this structure here, this is going to be called glycogen. And glycogen is the stored form of carbohydrate within our bodies. You can find it in the muscles, the skeletal muscles. You can find it within the liver. And it's going to be used as a source, a storage of carbohydrates. So whenever we need to, increase our blood glucose, we're going to break down these bonds within that glycogen in order to get some glucoses. So next we'll talk about lipids or fats. And lipids are not only used as a source of stored energy, but they are also important for the formation of certain hormones. They'll be used in the membranes of our cells. And so they come in many different types, including fatty acids, triglycerides, phospholipid steroids, and eicosanoids. First, talking about fatty acids and triglycerides. So fatty acids are going to be, the building block for lipids. And so here you see a saturated fatty acid versus a unsaturated fatty acid. So the saturated are that like palmitic acid, or just going to be the straight carbon backbone here, while in the unsaturated, you see that little kink that occurs in the skeleton, changing the structure of the fatty acid. And so these fatty acids are actually bound together in groups of 3, and that's called a triglyceride. And so what it is, it's 3 of these fatty acids with a glycerol backbone, which is a sugar. Okay, so 3 of these fatty acids are put together to form triglycerides, and this is the way that we store lipids in the body, through these triglycerides. And so it just doesn't need to be 3 of the same. It doesn't need to be 3 saturated or 3 monounsaturated. They were just all bound together through this carbon backbone attached to that glycerol here, in order to store fats. Here we see an example of another type of lipid, and it's called a phospholipid because it has this phosphate head, which is polar, right? Remember, polar meaning that it's going to have a charge up top. And then it has the fatty acids, which are non-polar. And so it's this combination of a polar side here in the head, and then these non-polar tails, which makes it great for the structure of our cellular membranes. And so to the outside, facing the extracellular fluid, and then facing the inside or the cytosol of the cell, you're going to find these polar heads that are going to interact with the aqueous solutions on the outside of the cell and the inside of the cell. And then in the middle, we're going to have this barrier of these non-polar tails that are going to come together. And this is going to make a great structure, a great wall for the cell. And it's going to prohibit just any molecules from coming across within the cell. And so these phospholipids, they're very, very important because they have a quality, and this is an important term, called amphiphatic, meaning that they possess both the qualities of polar and nonpolar, meaning that the polar head is hydrophilic, the tails are hydrophobic, and since they possess both those qualities, like an amphibian, right? They could go in the water and out of water. That's called amphipathic. Here we see lipids used to form major hormones in our body. So, again, a lot of times in health and fitness, fats get demonized, carbohydrates get demonized. They all have their importance within our bodies. And now, again, it just when you come to health and fitness, it's just how much do you need in your diet based on what your goals are, right? But here we just see the importance of lipids in forming different hormones. So, here we have cholesterol, estriadiol, testosterone, very important, cortisol. So these lipids are going to be used to form, the backbone for these signaling hormones. The last lipid we'll discuss is eicosanoids. They are derived from these 20 carbon fatty acid chains called arachidonic acids. So these include prostaglandins, which are important for the inflammation and signaling when there is damage in our bodies, thromboxanes, which are important for clot formation, and then leukotrienes, which also play a role in inflammation signaling as well. So we've talked about carbohydrates, we've talked about lipids, now we'll talk about proteins. And I think when most think about proteins in the general population, they think about skeletal muscles and how much protein they have in their diet and bulking. But when you think about proteins from a physiological aspect, there are tens of thousands of different proteins in all the different cells of your bodies. And they each have different structures and they each have different functions as well. And so they're going to carry out multiple events within our cells, and they're very important. And we produce all these proteins to carry out different tasks. And so the different functions could be structural or regulatory, such as hormones, contractile, like our skeletal muscles and our heart, immunological, to help fight off different things within our body, to help transport materials, or catalytic to help carry out different reactions. And so all of these different types of proteins are going to play a significant role within our cells and within our bodies. And so monosaccharides are the building blocks for carbohydrates. You have fatty acids, which are the building blocks for triglycerides and other lipids. But then you have proteins, and the building blocks for that, which most people know, are amino acids. There are 20 of them. They each have this carbon backbone here that is included with a carboxyl group. The carboxyl you see here, the carbon attached, double bonded with an oxygen, and then also bound to the hydroxyl group as well. That's your carboxyl group. You will have the amino group, where the term amino acid comes from, the nitrogen bound to either 2 or 3 hydrogens here. And then you're going to have a side chain. And those side chains are what separate the 20 different amino acids. Some, because of those side chains, some have different characteristics. Some are non-polar, some are polar, uncharged, some are polar charged. So all of these different amino acids are going to be, with their characteristics, are going to be used to build up different kinds of proteins. And so once you combine these amino acids in different sequences, it creates different structures, and then that's going to lead to different functions. Similar to the carbohydrates, we can build up amino acids to form peptide bonds, and then multiple amino acids can be put together to form polypeptides. And so the process is the same. We're going to use dehydration synthesis to combine 2 amino acids, and then you're going to get a dipeptide with the water as one of the byproducts, right? And in the same way, we, can use 2 amino acids and build them up through an anabolic reaction to get a dipeptide. We could use that water to then cleave these dipeptides through hydrolysis to get to single amino acids. And so this dehydration synthesis and the hydrolysis, either building up or breaking down monomers and dimers and polymers, we're doing this because we need to carry out these different reactions. Sometimes we're going to need energy, so we're going to break apart bonds to get that energy. Sometimes we're going to need those building blocks. So again, if we take a protein and cleave it and slice it down, we can have those amino acids to then build other proteins. But then sometimes too, we need to put them all together. So it's these constant reactions from building up and cutting down and breaking down in order to get the, function that we need. So here we see that proteins are organized in 4 different structures. The first or primary structure that we see is just a polypeptide chain, which is just multiple amino acids bound together. Just right here as it's shown in a straight line. And then once we start to build upon that polypeptide chain, all these amino acids are going to react with one another to either form what's called an alpha helix, as you see here, or a beta sheet. And both the alpha helix and the beta sheet are the secondary structure of proteins. These, multiple alpha helixes and multiple beta sheets will come together to form more of a 3D structure called a tertiary structure. So here we see both the combination of these alpha helixes and these beta sheets. And then once we start to add these polypeptide chains together, that's where we're going to get our quaternary structure. And so we're actually going to get these polypeptide chains coming together, and we're going to form this protein. We're going to form the structure of this protein, and then it's going to carry out its function once it's complete. An important reason why we like to keep this homeostatic environment, the same temperature, the same pH, is because that this baseline temperature and this baseline pH is when these proteins in our body react the best. They're going to carry out their reactions in the most optimal environment. And we're going to talk more about that in metabolism, but that's why we set this environment, just to make sure that these proteins can behave properly. And what happens is if there's too much acids introduced or the temperature gets too hot, too cold, proteins can then lose their shape, and this is called protein denaturation. So again, when proteins are put in conditions outside of their optimum, that's when you're going to see the protein become denatured. And it's very important to point out that the structure of the protein defines the function. And so, like I said, we have tens of thousands of different proteins, and each have isomers as well. But those shapes are very important for what that protein does, as we'll see here in a little bit. And so once that protein becomes denatured, it messes up the function, and then it can't behave properly. Many different proteins will bind to other molecules, and these molecules or ions that bind to proteins are called ligands. And so you'll see that sometimes ligands will turn on a protein. Sometimes they'll turn off a protein. They have many different functions, and a lot of times these ligands will change the structure and therefore change the activity of a protein. And these ligands are going to bind to sites, conveniently enough, called binding sites, which is the region of the protein where the ligand binds. So you're going to have different things that affect the ability for a ligand to bind to a protein. And those 3 are going to be called specificity, affinity, and saturation. One thing that affects the binding of a ligand to a protein is specificity. And that's going to mean that these proteins have specialized sites, specialized structures for these ligands to bind. And so to prevent, let's say, ligand 1 over here from activating protein A and protein B when it's upregulated, when it's floating around in the cell or in the bloodstream and then is coming in close proximity, that specificity is going to make sure that this protein is only, let's say, activated or modulated when that ligand comes into that binding site of protein C, because that binding site, its structure, is specifically made for that ligand. Affinity is going to be the strength to which a ligand binds to a protein. And so a lot of times the affinity is based on the charges or the composition of both the ligand and the protein, and that affects the ability of that protein to bind to the ligand. And so if you have a protein with two close by ligands, well, certain times the protein may be able to bind to both ligands, but the one that it has a stronger affinity to is going to be the one more likely that binds. And you also have different case scenarios where a protein may not have a strong affinity to a ligand, so therefore it's less likely that that ligand will bind to the protein. Next, we have saturation. And saturation is going to be the degree to which the binding sites of a population of a protein molecule are occupied by a ligand. And so the more ligands you have, being circulated, the more likely it is they're going to bind to these binding sites. And so a lot of times, that's how we either upregulate or downregulate. If you think about hormones, if you think about other messengers in the body, well, if you want to increase the likelihood of that ligand binding to a protein, you're going to make more ligand. If you want to decrease the likelihood of it binding to that protein, you are going to decrease, right? And so when it's highly saturated, when there's a lot of ligand floating, you're going to reach your binding maximum at some point. And so again, it's just based on the number of ligand binding to that protein. We have other forms of regulation when it comes to ligand protein binding. We have what's called allosteric modulation, which is a direct influence, and then a covalent modulation, which will influence one or more proteins to bring about an action. Allosteric modulation is when a chemical substance known as an allosteric modulator binds to a site called the allosteric site, which is separate from the binding site. And so it could either activate or inhibit the ligand from binding to the binding site. If it is an allosteric activator, it's going to bind to the protein, and that is going to change the structure of that protein to then allow the ligand to bind. An allosteric inhibitor is also going to bind to an allosteric site, change the structure of the protein, which then prohibits that ligand from binding to the protein. Covalent modulation is going to differ from allosteric modulation. In allosteric modulation, we had something binding to a specific site in order to affect the ligand binding. Here we are going to have an addition of a molecule to one or more amino acids in order to change the structure of the protein to allow for changes to the binding site. So here we see a phosphate group added to the protein. This is going to take place on one or more of the amino acids. And again, when that phosphate group is added from another molecule, it's going to change that binding site, and in this case, allow for our ligand to bind to the binding site. Nucleic acids are repeating chains of nucleotides, and they're going to be the basis for DNA and RNA, which helps store our genetic materials. And these genetic materials are going to be used to make proteins, and we're going to be discussing that a little bit later on in this lecture series here. But they can also be used for components of high energy molecules, which could be cleaved, and then again release that energy to carry out different chemical reactions. So nucleic acids have that nitrogen base, typically around a single or double ring. They have a pentose sugar, which again we said earlier, that could be the deoxyribose or just ribose. And then they also have a phosphate group, which alternate with those pentose sugars, and that's going to form the backbone of these nucleic acids. Here we're going to break down nucleic acids a little bit more. We see again that phosphate group bound to the pentose sugar, which is either deoxyribose in DNA or ribose in RNA. And then we have these nitrogenous bases. We have cysteine, thymine, adenine, and guanine, which are found in DNA. Cysteines are going to bind to guanines. These two will always bind to one another. And then thymines are always going to bind to adenines in DNA. Except for in RNA, the only difference that we're going to have is thymine is going to be replaced with uracil. And so, again, we're going to talk more about genetic expression at a later date. But ACGT is going to be found in DNA, and then ACGU will be found in RNA. Here we see, again, just the structure of DNA in this double helix structure here that we see. Again, the adenines are going to bind to thymines in DNA, guanines are going to bind to cytosines, and then we're going to have just 3 billion base pairs of these all in a row in every single one of our cells. So all of this genetic material is stored within those cells and then at some point expressed in order to create and synthesize proteins. Last, we are going to discuss adenosine triphosphate, otherwise known as ATP. And this is the energy currency of our cells. This is how we are going to store and transfer energy within our cells to carry out chemical reactions. And so the name comes from the combination of adenine and the ribose, which gives us adenosine, and then these 3 phosphate groups, adenosine triphosphate. And once we cleave one of these phosphate groups, it's going to release energy, and then that energy can be used to carry out other functions within the cell, to carry out other chemical reactions in order for the proteins to carry out certain functions. And so we need energy for the cell to live. And so when we break down food and we consume energy through food and liquid, this is what eventually we are going to get. This is the most basic form of energy transfer within our cells. And so when we cleave one of these phosphate groups off, that's going to give us adenosine diphosphate. If we cleave another one off, adenosine monophosphate, AMP. And so this is how we are going to transfer energy within our cells. Thank you so much for watching the part 2 video on chemistry for human physiology. Please check out the other lectures where we will be discussing other aspects of human physiology, health, and fitness.